Good morning. It's April 16th, and this is your daily brief in science. Here's everything you need to know. Amazon Web Services is launching an AI platform for drug discovery called Amazon BioDiscovery, offering researchers a library of biological foundation models that can be fine-tuned for specific projects and guided by an AI agent that selects models and evaluates synthesis candidates for a fast lab-in-the-loop cycle. Lab partners test the most promising antibody candidates and feed results back to scientists, creating a feedback loop that refines design iterations. The platform uses a subscription model with a 5-experiment free trial and avoids ads in search results. Collaborative workflows encode decisions on model chaining, data processing, and quality thresholds to continually improve models and shorten queue times. It enables parallel experimentation and automates parts of the analysis to reduce manual intervention. Results flow back into the platform to compare predictions with actual outcomes, driving iterative refinement of workflows. Analysts say queued projects can move forward immediately, and computational biologists can support more programs with improved workflows. Computational biologists can build reusable, no-code workflows, while bench scientists run multiple experiment variants with agent-guided inputs, boosting collaboration and speed. AWS's Luca Giancarlo highlighted the developability benchmark as a key measure for evaluating model suitability and performance. The platform emphasizes data isolation and ownership, allowing researchers to train custom models on their own data with private fine-tuning and in-house deployment options. Researchers can train models on their data to improve predictions while maintaining privacy and ownership, with a companion article noting AI's potential to transform drug testing workflows, though technical specifics aren't detailed in the source. A new Cochrane review pools 17 studies with over 20,000 participants and finds that anti-amyloid Alzheimer's drugs deliver only trivial cognitive and dementia improvements after about 18 months, with benefits not meaningfully felt by patients or their caregivers. The review covers drugs such as lecanemab and donanemab and notes little to no clinically meaningful benefit in early-stage Alzheimer's, while carrying higher risks of brain swelling and bleeds. Critics say pooling older, ineffective trials with newer ones may bias conclusions, though proponents argue all trials target the same amyloid removal goal. Public health implications loom. Treatment costs run roughly £50,000 to £80,000 per person annually, and MRI monitoring is required, affecting NHS adoption and planning for future therapies. In the EU and Germany, reimbursement decisions by bodies like the GBA and GKV Spitzenverband influence whether manufacturers continue marketing these therapies, while regulatory bodies such as NICE question cost-effectiveness given uncertain and modest benefits. Regulatory approvals for aducanumab and donanemab are controversial, with calls for longer follow-up and more diverse populations in future research. German and other regulators express skepticism about added clinical value, aligning with findings that these antibodies do not outperform symptom-focused or older treatments in practice. Researchers emphasize that observed effects are too small for real-world notice and note a heavy treatment burden from visits, infusions, and MRI monitoring. The 18-month window is acknowledged as short for a slowly progressing disease, and real-world use could involve longer treatment, potentially shifting risk-benefit calculations. Clinicians and charities urge nuance, noting that some analyses mix failed trials with newer ones and that individual drugs may differ in effect, warranting cautious interpretation. Some experts caution against broad dismissal of anti-amyloid therapies, pointing to methodological limits in the existence of drugs with different mechanisms not covered by this review. A rising national threat from drug-resistant Shigella infections is under scrutiny by the CDC, with no FDA-approved oral treatment currently available. Shigella spreads mainly through fecal-oral transmission, sexual contact, and contaminated food or water, and symptoms typically include diarrhea that can be bloody, fever, and stomach cramps, starting 1 to 2 days after exposure and lasting 5 to 7 days. Most cases resolve without treatment, but antibiotics are used for severe illness or to curb transmission in high-risk settings, and prevention hinges on rigorous hand hygiene and avoiding contaminated water and crowded environments. 2 Pennsylvania Department of Health researchers contributed to the CDC study, while state-specific numbers for Pennsylvania and other states have not been released. The data show most affected individuals are adult men with a median age around 41, and among travelers, a large majority did not travel internationally recently. Georgia notes its health department does not track exact annual Shigella numbers within the state. Young children, especially those in childcare, people in crowded living conditions, and individuals with limited access to clean water and toilets are among the most at risk. Preventive measures emphasize thorough hand hygiene, safe food and water practices when traveling, avoiding sexual contact with sick individuals, and for travelers to certain regions taking precautions to avoid contaminated foods. Pennsylvania's guidance calls for rigorous hand hygiene, careful diaper handling, disinfecting changing areas, preventing diarrhea spread in childcare, supervising children's hand washing, and avoiding food handling by those ill. Travelers are advised to boil, cook, peel, or avoid certain foods in certain regions. New Jersey guidance includes thorough hand washing, disinfecting diaper changing areas, avoiding swimming while ill, and consuming only treated or boiled water and cooked foods when traveling to developing countries. Most XDR Shigella cases have occurred in adult men, with about 1/3 of patients hospitalized, reflecting a shift from earlier outbreaks that affected children. A landmark ancient DNA study of roughly 16,000 West Eurasian genomes covering more than 10,000 years finds about 479 genetic variants with strong signals of directional selection since farming began, challenging the idea that recent human evolution was rare. The research shows widespread adaptive evolution in the last few millennia, with selection intensifying after the Neolithic shift to agriculture. Many strongly selected alleles tie to present-day traits and diseases, including lactose tolerance, skin and hair pigmentation, autoimmune risks, and neuropsychiatric traits, though interpretation must consider pleiotropy and other confounding factors. Some variants linked to disease risk or complex traits, such as multiple sclerosis, cognition, and mental illness, show frequency changes whose meaning is still debated. The study engages with broader debates on polygenic adaptation and complex trait evolution, referencing prior work on disease pressures and pathogen interactions. Findings have implications for medical genetics and precision medicine by highlighting recently shaped variants that may influence disease risk and therapeutic strategies, while situating clinical work in an evolutionary context. Historical examples of selection, such as CCR5 delta 32, and the roles of geography and demography are discussed to frame how adaptation arises across populations. Immunity-related genes emerge as major targets of selection, with notable shifts in variants affecting tuberculosis susceptibility and HIV resistance across periods. The methodologies could extend beyond humans to areas like agriculture, conservation, and climate resilience by identifying adaptive alleles in domesticated or wild species. Strongly selected alleles also relate to metabolic and neuropsychiatric traits, including type 2 diabetes risk, autoimmune conditions, and schizophrenia or bipolar disorder, though causal links must be interpreted cautiously. The evolution pattern is complex, with many allele frequency changes resembling roller coasters driven by population movements and drift rather than steady directional increases. A new high-resolution atlas maps human liver zonation at a 2-micron scale, revealing patterns of gene expression unique to humans across liver zones. Researchers combined multiple spatial omics technologies, 10x Visium, MERFISH, snRNA-seq, and phenocycler imaging to chart hepatocytes and non-parenchymal cells, validating results through cross-method concordance and a web exploration tool. The study finds Kupffer cells relocate toward the lobule center in humans, indicating an evolved immune turnover mechanism to address higher cellular attrition there. 8 healthy donor livers from altruistic living donation provided samples, with comparative data from mice and other mammals to contextualize human zonation. Early steatosis begins with lipid buildup mainly in the pericentral zone, and machine learning links lipid content per spot to disease initiation in healthy livers, offering a resource to study progression. Pericentral hepatocytes are enriched for xenobiotic metabolism, bile acid synthesis, lipid biosynthesis, and WNT pathway components, while periportal hepatocytes show immune-related gene enrichment and gluconeogenesis, including human-specific patterns like pericentral PCK2 and GLUT2. Context notes cover data licensing and access options for nature, and reference related liver biology literature and single-cell mapping resources. The atlas identifies spatially resolved adaptations in liver cells from individuals with steatosis, highlighting zone-specific changes. It demonstrates potential for tracing disease development and guiding targeted therapies, and suggests the approach could map other organs at single-cell resolution to transform human biology understanding. Liver histology from living donors shows clear transcriptional differences from adjacent normal tissue, underscoring the need for truly healthy tissue as a reference for atlas construction. A collaboration among Weizmann Institute, Sheba Medical Center, and Mayo Clinic yields the first genetic atlas of a healthy human liver at 2-micron resolution, revealing a more complex regional organization than previously thought. The work enhances understanding of liver architecture and zonation, with implications for liver diseases and personalized medicine. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.